Dear students, in this module, we'll look at how the coding process for amino acid actually works. So given an amino acid codon amino acid conversion lookup table, then how can you convert a DNA or an RNA sequence that you may have obtained from a DNA or RNA database? Let's see. As you know, the nucleotide codons for the RNA contain A, U, C, and G, and that they code for proteins. Moreover, multiple combinations of these codons can actually code for a single amino acid. Also, there are 20 different types of amino acids, so therefore there are much greater number of combinations for the codons. If you look at this chart, then you can assemble the amino acid symbol that will be encoded by any possible combination of the nucleotides. As an example, if you have the first nucleotide as A, the second as T, and the third as G, then you will be encoding for the amino acid methionine, which is actually also the start codon. Similarly, you can code for the stop codons, which in this case will be two different combinations formed by TAG and TAA. Therefore, you can assemble all possible combinations for start, stop, as well as the amino acids that will form the protein. Now let's take an example of how this actually works. So if you obtain an RNA sequence, or for that matter a DNA sequence from a database, then you can start by looking for the start codon. The start codon, as we just saw, was denoted by ATG. So while you are going through the RNA sequence, then you can look at AT and G, and the place where it occurs, you can encode a methionine for this codon, and you can start collecting codons of 3 nucleotides following this, such as that, and that, and that, and so on and so forth. So for each of these codons, you look up your table and you replace it with the protein amino acid. Once you reach the end codon, the stop codon, then you can look at what is the entire chain of amino acids that is formed. Given below is one such example. So this DNA sequence is actually coding for this protein. And we have started from here, the AT and G, and stopped at TAA. So this is how you can extract the proteins that can be coded from an RNA or a DNA molecule. The only thing that you need to remember is that there is one start codon and 3 stop codons. With that information, as well as your lookup table, you are ready to go. Dear students, there is an important thing that you need to consider at this stage. What if there is a random combination of these nucleotides that you are searching for the start codon and it occurs somewhere in the sequence? So you can actually have multiple start codons. So you need to know which start codon is valid. Similarly, the stop codon can have the same occurrence, a random occurrence. So how will you find that it's not the valid stop codon and is just a random one? So, moreover, how do we select which stop codon to use from the 3 available options? So these are the points to ponder for you.